Hello everyone, welcome to Get My Class. So let's solve this question here. It says identify A, B, C and D. And here they have given a picture. They have mentioned A, B, C and D, which we have to identify. And they have given A, B, C, D as the options here, where A means operator, B is RNA dependent DNA, and C is sigma factor, D is RNA dependent RNA. Option B says A is promoter, B is DNA dependent RNA, C is Rho factor, D is DNA dependent RNA. Option C says A is promoter and B is DNA dependent RNA, C is sigma factor, D is DNA dependent RNA. Option D says A is operator, B is RNA dependent DNA, C is Rho factor, and D is RNA dependent RNA. So let us understand the question. It says identify A, B, C and D. So we have to identify A, B, C and D. But before we identify, let us actually identify which picture is this. What is it about? So first one, it says initiation. Of course, they say initiation. What have they given? They have given double helical structure of DNA. And here they have mentioned sigma factor and they have mentioned about the polymerase. So which type of polymerase is it? That we have to identify. Okay. And this particular A, they are actually showing up to this region where the sigma factor binds. If you don't understand like what are they talking about, let us go to the next step, that is elongation. Here you can see that there is synthesis of RNA, right? So as soon as you see that there is synthesis of RNA, so which process is this? This is nothing but transcription. And what is transcription? Transcription is the process of synthesis of RNA. Yes? Yes. Now you're sure that it is transcription. Let us go back to the first step or the initiation. So in the initiation process, what happens? The sigma factor, okay, it identifies a particular sequence of the DNA, which is called as the promoter sequence. What is this? Promoter. So sigma factor will identify a promoter and it will help the RNA polymerase to bind to the promoter. So A is what? Promoter, and B they have mentioned as polymerase, but what type of polymerase? Here the polymerase is of course RNA polymerase, but it is DNA dependent RNA polymerase. Why DNA dependent? I'll tell you. DNA dependent RNA polymerase. Okay, polymerase they have already mentioned, but still I will mention it here. So this is this orange color, whatever they're showing, that is DNA dependent RNA polymerase. Why? Because it is dependent on the DNA sequence so as to synthesize the RNA. Okay, so let us understand here. So here the DNA acts as a template, right? DNA is acting as template. DNA. And based on the sequence on the DNA only, RNA will be synthesized because RNA will be complementary to the template strand. Okay, yes. So that is why this polymerase is what? It is DNA dependent RNA polymerase. Okay. So A and B we have done. That is A is promoter, B is DNA dependent RNA polymerase. So now C. So Rho factor initially it binds and then further it not Rho factor, sigma factor. Sigma factor it binds to the promoter and further it will come out. Like once it begins its synthesis and elongation occurs, then it doesn't need now. Its work is done. So this C is nothing but the sigma factor. What factor is this? Sigma factor. It moves away. So this is A, this is B, and this is C, sigma factor. Then further, here they have mentioned as D. So when sigma factor moves away and RNA polymerase will continue the synthesis of RNA. And then this is the terminator sequence. This is the terminator sequence where the RNA polymerase has to stop its transcription and move away. And that is done by the help of the Rho factor. So Rho factor will help, it will bind to the terminator sequence and help in termination of the transcription. Okay, so that is Rho factor. Okay, and here they have mentioned D. So what is D again? D is nothing but the polymerase only. Which polymerase is this? This is nothing but DNA dependent RNA polymerase. So it will still remain the same polymerase, right? DNA dependent RNA polymerase. Why is it DNA dependent? I have already told you, because it is dependent on the DNA sequence or the template of that is DNA template, which acts as you can say template for the synthesis of mRNA. Okay. So now option A says A is operator, which is wrong. It is not an operator, it is promoter. Okay, hence here itself you can rule it out. Option B says A is promoter, which is correct. Correct. B is DNA dependent RNA, that is also correct. C is Rho factor, they say. So our C is sigma factor and not Rho factor. Hence it is ruled out. Option C is A is promoter, correct. B is DNA dependent RNA, that is also correct. So this is A, this is B. And C is sigma factor, that is also correct. D is again DNA dependent RNA, that is also correct. So option C should be the correct answer. Option D says A is operator, which is again incorrect. So what is the correct answer here? The correct answer is option C. I hope you understood the explanation. Thank you.